Hello, welcome to another screencast where this time we'll be talking about just an overview of some of the biological macromolecules. So we want to be able to describe composition of the macromolecules and focusing specifically down here, just looking at the quick overview of the classes of the biological macromolecules, and then looking at the monomers and then the polymers of each. In other videos, we'll go into each of the four different kinds there. So again, following this kind of reductionist view of life and looking at the cell as the basic unit of structure and function, looking at cell functioning has a lot to do down here with what's going on at the molecular level. So when we talk about the molecules, macromolecules, we're talking here they talk about DNA, or they're pointing out specifically the molecule of DNA. And so the DNA is, of course, a nucleic acid. We also then have proteins and carbohydrates and lipids. And so we'll address each of these four macromolecules briefly here in turn. So in other videos, we talked about how to make these macromolecules from smaller individual monomers, right? So going from individual monomers to larger polymers and then back again. So from these larger polymers into individual monomers. So this pattern somewhat holds for all those different classes of macromolecules. So we can look at this at the monomers for nucleic acids, the monomers for proteins, the monomers of carbohydrates, and to an extent the monomers of lipids. And then we can also look at each of those as a kind of polymer as well. And so cells have the ability to create larger polymers from these individual monomers, and then in reverse doing that, hydrolyzing those polymers down in individual monomers. So if we look at the first class here, just in general, the nucleic acids are polymers of nucleotides. So the nucleotides here would be the monomer. It's supposed to be an N. Would be the monomer units. And these are the nucleotides here. And you would assemble those nucleotides together in a chain to create some sort of polynucleotide. Right? So many of them would be a polynucleotide. And so DNA can be considered a polynucleotide, right? So here we see this polynucleotide or nucleic acid. Unfortunately, in biology, sometimes things have the same molecule as more than one name, which is frustrating sometimes for first-time learners or even second-time learners of this. But here you go. So these nucleic acids are made up of these individual chains of monomers here. There's a nucleotide, there's a nucleotide, there's a nucleotide. And you see that this nucleotide number 1 here is bonded to this nucleotide number 2 by this bond right here. And then we see it again here. So this is the result of a condensation reaction. And if we were to go the opposite way to take this polymer and break it down into these individual nucleotides, that would be a hydrolysis reaction. So more videos on the details of this DNA and RNA later, but the nucleic acids are the first class of these macromolecules. And not only do we have DNA as a nucleic acid, you also have RNA as a nucleic acid, and these are primarily single-stranded. And again, the function of most of these nucleic acids is information storage. So in terms of like hereditary information, but then in the case of some of these RNA molecules specifically, and even some DNA molecules, you have a little bit of functionality, right? And we'll get into this detail later on when we do some of the RNAs in terms of some RNAs having sort of catalytic properties to them. So nucleic acids, first category of those macromolecules. Sort of a parallel to that is another molecule that many biology learners are familiar with, and that is ATP. And we can see ATP as a modified kind of nucleotide where this would be the nucleotide here, the phosphate, the sugar, and the nitrogenous base. But then you add a couple more phosphate groups on there, right? So this is, in my opinion, sort of an example of the cell reusing molecules for different purposes, or repurposing, I guess I should say, instead of reusing, repurposing some molecules for different functions. So again, we'll see this a little bit later, but again, that ATP molecule related to those nucleic acids, or at least the monomers of those nucleic acids. Moving along, the second group here are the proteins, and proteins are the workhorses of cells. They have lots of roles in functioning, in terms of structuring, in terms of signaling, and so we will see proteins a lot, and we'll have a lot of videos on a handful of videos on protein structure and protein function. But all of the proteins are made of these structures right here that are the amino acids. So the amino acid is the monomer of any protein, and there are 20 different amino acids, and the way that you string those amino acids together, right, so this would be an amino acid, this would be a monomer bonded to this next amino acid right here by this peptide bond to create a dimer, and so on and so forth. And so I can build these proteins, these polymers, by stringing together amino acids all along the way there. So much like polynucleotides and nucleic acids are the same thing, sometimes you'll hear, I'll just talk about polypeptides and proteins. And so proteins, polypeptides, for the most part, are interchangeable for us, and those are the polymers of the monomer amino acids. And again, you can go back and forth here. So this arrow is showing you making the polymer, but you could go backwards from that as well. And again, we'll do more videos on this as well, but here we see just the early stages of sort of protein structure, where we have this chain of amino acids, and then from here over, this chain of amino acids will fold, right? And this folding is correlated then with functionality, and we will see this again in other videos as well. But nucleic acids, proteins, are two macromolecules that are made from smaller individual monomers to get a larger polymer. This happens in carbohydrates as well, and the monomers of carbohydrates are things like glucose, which is a monosaccharide, or a single sugar. And if you take something like glucose and you bond it to another glucose that they're showing you here, so this would be a condensation reaction, you end up getting a disaccharide such as maltose. You get a disaccharide like maltose there. So if you took a glucose monosaccharide and a fructose monosaccharide and condensed them together, you'd get this disaccharide, which is just your regular table sugar, right, which is sucrose. So if you're eating table sugar, sucrose, you would digest it going this way, and this would be a hydrolysis reaction, and you end up getting these individual monomers here, monomer and a monomer. But if you're going this way, you get, in this case, you'd get a dimer, and not necessarily a polymer. So, but the pattern is still going to hold here in terms of putting monomers together to get larger structures that have different properties to them. If you string a whole bunch of glucoses together, where each of these little green hexagons is supposed to be a glucose, you get different kinds of polymers, and these are, again, the polysaccharides, of which starch is one and glycogen is another. And then this down here is showing you what cellulose looks like. And so again, the theme here is that I have a whole bunch of individual monomers that have been all bonded together in a very specific way to get me a different kind of polysaccharide, which is going to have different but similar properties to one another. And again, more detail on that in the carbohydrates videos. So nucleic acids, proteins, carbohydrates, And then lastly, our final group of macromolecules are the lipids, which don't fit but kind of do some of this monomer into a polymer situation. Here we have a triglyceride. So down here is a triglyceride, which is kind of a polymer of these fatty acids and then a glycerol molecule. So it's 3 of these fatty acid molecules bonded to this glycerol gets you a triglycerol. And these would be the molecules like that are for fats and oils. And again, we have videos on lipids and talking about saturated versus unsaturated fats. So more on that later. But again, these lipids in this case are primarily energy storage molecules and potential energy storage in all of these bonds of these fatty acid tails here. So these are all potential energy sources, and cells use these primarily that way. There's another example of lipids, and again, we'll go into this in more detail, but these phospholipids, and we see phospholipids, which are modified triglycerides, to have this phosphate, including head here, that is polar. It's charged up here on this head, so this allows us to allow cells to have membranes that are going to separate one aqueous environment from another aqueous environment there. So we see lipids not only as energy storage molecules, but modified ones as membranes, cell membranes, organelle membranes, etc. And then lastly, you have the steroids, which don't fit sort of any of the other categories, but you'll know a steroid here by this classic kind of like honeycomb pattern here. And then some examples of these. So cholesterol, we'll see that in different capacities in terms of the membrane, but then also you're looking at hormones here. So these are classified as lipids, and so these are involved in sort of cell signaling pathways as well. So, right. So that was a very quick blitz through the classes of the biological molecules. So there are 4 of them. Again, the nucleic acids, the proteins, carbohydrates, and lipids. Quickly talked about monomers of each of those, and then some of the polymers as well. Probably a good idea to pause and go back. But again, just a quick overview and primer. If you have any questions, write them down, bring them to class, and we'll see you there. Thanks for listening.